section E. Okay? The graph given below, that is we're talking about this graph, okay? We're talking about this graph, is representing the reproductive cycle in females. What is that? The menstrual cycle. Days of female sexual cycle, we know it takes 28 days to 30 days, 1 month. Analyze the graph in regard of a normal menstrual cycle and answer the questions given below. Very good. Identify the hormones involved in the menstrual cycle. See, there are basically 2 hormones which are involved in the menstrual cycle. One is the luteinizing hormone and the other one is the follicle stimulating hormone. Okay? The follicle stimulating hormone. Now we see, explain the ovarian phases during the menstrual cycle. Okay, so what is the phase of the the menstrual cycle? What is the phase from the 5th day to the 12th day of the cycle called as? Right? What is the phase 14th day of the cycle called as? And what is the phase 16th day to 25th day of the cycle, that is the last phase of the cycle called as? Good. What influence does these hormones have on different ovarian events labeled as A, B and C, that is in these? Or you have an option here. Male and female gametes in human beings differs from each other in both structure and function. Yes. I'm sorry. Enumerate some major differences between the male and the female gamete. Okay, so we are going to talk about the first question, and let us see what the first question demands from us and what the what the question is wanting. So first subpart of the question, identify the hormones involved in the menstrual cycle. Now my dear friends, as you can see this lower line, which is a very dark line. Forget about if I don't know whether you can see it as a dark or a light line. Follow this line I'm talking about. Okay, you can see with the finger. So this line is going right above peak. This line is taking the peak at the 14th day, that is the mid of the cycle. This line is taking the peak. Okay, and here you see that this line is coming here and then going like this, right? So we can say that identify the hormones involved in the menstrual cycle. The hormones involved in the menstrual cycle will be these 2 lines representing the follicle stimulating hormone and the luteinizing hormone. Okay, so the first answer to these will be the hormones involved, the hormones involved in the menstrual cycle will be what? This will be the follicle stimulating hormone, follicular stimulating hormone FSH, and the second one will be the luteinizing hormone LH. Okay, so these two hormones are involved in the menstrual cycle. One is the follicular stimulating hormone and the other is the luteinizing hormone LH. Right? Now I'll rub this and let us move to the second part of the question. The second part of the question says that explain the ovarian phases. Okay, explain the ovarian phases during the menstrual cycle. So it is asking 5th day to 12th day of the cycle. The 5th day to 12th day of the cycle, that is the first phase of the cycle, is also called as the follicular phase or the proliferative phase. Okay, the first, so the answer would be A. The second answer A, 5th to 12th day, 5th day to 12th day is also called as follicular phase or the proliferative phase. Okay, the follicular phase or the proliferative phase. The second part of the cycle, that is from the 14th day of the cycle, the 14th day of the cycle, where the luteinizing hormone will be at the max, called the LH surge. This phase is called as the ovulatory phase, where the release of the ovum will be taking place. Okay, so the B, the 14th day, is called as the ovulatory phase, where the release of the ovum will be taking place. And finally, the 16th to the 25th day of the cycle. The 16th to 25th day of the cycle, that is the last phase, this last phase of the cycle, this is called as the luteal phase. Okay, this is called as the luteal phase. Luteal, luteal phase. So even the second part of the question is over. That is, yes, the first part is called the follicular or the proliferative phase. The second part, that is the 14th day of the cycle, is called the ovulatory phase, and the third part is the luteal phase from the 16th day to the 25th day of the cycle. Right. Let us move on to the third question, that what influence does these hormones have on different ovarian events labeled as A, B and C. Okay, so what this question wants, that what influence does these hormones have on different ovarian events. Okay, so what is the effect of the follicular stimulating hormone and the luteinizing hormone on these three different phases, which we will discuss now. So during the 5th day to the 12th day of the menstrual cycle, we see that the follicular stimulating hormone is the most active. Okay, the follicular stimulating hormone is the most active during this phase. This follicular stimulating hormone is released from the anterior pituitary. Okay, it is released from the anterior pituitary, and this follicular stimulating hormone, this will stimulate the ovarian follicle to release what? To release the estrogen, to release the hormone estrogen, and this hormone estrogen will, this will help in the differentiation of the endometrium lining of the uterus. Got it? I'll write this down and then explain you once again. So I'll write this as, this is third part, okay, third A part. Follicle stimulating hormone is secreted by anterior pituitary. This stimulates ovarian follicle to release, okay, to release the hormone called as estrogen, to release the hormone estrogen. to release the hormone estrogen, which causes differentiation, okay, which causes proliferation, differentiation, proliferation of endometrium, Okay, so FSH is secreted by the anterior pituitary, right? By the anterior pituitary, thus stimulates the ovarian follicle to release the hormone estrogen, and this hormone estrogen will cause the proliferation of the endometrium. So did you get the point? From the anterior pituitary, the hormone called follicle stimulating hormone is secreted during the first phase of the menstrual cycle. What this will do, this will stimulate the ovarian follicle, and on stimulation the ovarian follicle will stimulate the hormone called estrogen. This estrogen will cause proliferation of the endometrium. Got it? Now let us see the second part. That is, what is happening during the 14th day. In the later phase of the menstrual cycle, what is happening? Both FSH and the luteinizing hormone are at their, they are high. On the 14th day, we will see that the luteinizing hormone is at its peak. The luteinizing hormone, it is at its peak, and this will, this peak of the luteinizing hormone is called the LH surge, which will stimulate the Graafian follicle to release the hormone called the progesterone, and it is this hormone progesterone which maintains the endometrium lining of the uterus. What was estrogen doing? Estrogen was proliferating the endometrium lining of the uterus. And what is this hormone progesterone doing? Progesterone is, is maintaining that endometrium lining. Okay? So I'll write this down again as point B, that both LH and FSH attain peak level at about 14th day. Attain peak level at about 14th day. Rapid secretion of LH. The rapid secretion of LH is called LH surge, which induces, which causes, release of hormone progesterone from the Graafian follicle, Okay? So both LH and FSH attain the peak level, at about 14 day. This rapid increase secretion of LH is called the LH surge. This is called as the LH surge, which causes the release of hormone called the progesterone from the Graafian follicle. And what does this progesterone do? I'll write it here. This progesterone, the progesterone maintains the endometrium lining of the uterus, maintains endometrium lining of uterus. Okay. So the progesterone maintains the endometrium lining of the uterus. Now let us come to the third part, 16th day to 25th day of the cycle. Now what is happening here? What is happening here? The remainder of the ovarian follicle is being stimulated. Okay, this is being stimulated by the LH to form the corpus luteum. Okay, to form the corpus luteum, and actually, okay, I'm sorry, this actually forms the progesterone. Okay, this actually forms the progesterone which maintains the cycle. So I'll write that again. B, I'll write the B1 again. That 14th day of the cycle, 14th day of the cycle. Please rub what you have written. 14th day of the cycle, what is happening? Both LH and FSH are high. Okay, both LH and FSH are high, and increase in LH, increase in LH is called as LH surge. Okay, increase in LH is called the LH surge, right? This increase in LH, luteinizing hormone induces Graafian follicle. Okay, the mature ovarian follicle is called the Graafian follicle. Induces the Graafian follicle to rupture and release of ovum. That is ovulation. So please, I'm really sorry, please delete what you are, please rub the B part. A part was perfectly all right. Okay, this will be the B part, that is 14th day of the cycle. Both LH, luteinizing hormone, and the follicle stimulating hormone are high. There is an increase in the luteinizing hormone. This is called as the LH surge, and because of the increase in the luteinizing hormone, this will cause the rupture of the Graafian follicle, and hence the release of the ovum. That is, ovulation will take place at this stage. Now we see the C stage. What is happening here? In the C stage, that is the 16th to the 25th day, okay, the remaining cells of the ovarian follicle, they will get transformed into the corpus luteum. Okay, corpus luteum by the luteinizing hormone, and this releases the progesterone hormone which maintains the endometrium layer. Okay, 16th to 25th day, what we see, the remaining cells of ovarian follicle are stimulated by LH. Okay, the remaining cells of ovarian follicle are stimulated by LH to develop corpus luteum, which secretes progesterone, and it is this progesterone which, can this be seen? Okay, and this progesterone maintains endometrium layer. Okay, endometrium layer. Right? The remaining cells of the ovarian follicle are stimulated by LH to develop corpus luteum, and this corpus luteum secretes progesterone, and this progesterone maintains the endometrium layer. So friends, you write down all this, and this OR question, that is difference between the male and the female gamete, we will be discussing in the next session. Okay? Okay friends, so I hope you must have noted down. And now we will move on to the next question. Describe the process. Okay, the next question, I think we were discussing this over here, and we were, the OR question we didn't discuss. That is, the male and female gametes in human beings differ from each other in both structure and function. Enumerate some major differences between the two. Yes, the male gamete and the female gamete, the male gamete sperm, the female gamete the ovum. There are some differences. For example, the male gamete or the sperm is motile, the female gamete, the ovum, is non-motile, right? The male gamete, the maturation takes place about 64 days, and for the female gamete, the ovum, the maturation takes place about years. Okay, the male gamete is produced, that is, the sperm is produced in the testes. This female gamete ovum is produced in the ovaries, right? The male gamete body is differentiated into 4 parts: the head, the neck, the middle piece, and the tail. The ovum, the body, is not differentiated into any parts. Okay, so now we will see, we will write in a tabular way how these two can be differentiated. How these two can be differentiated. So I'll write here the male gamete sperm. Okay, actually there's the male gamete sperm, and here I'll write the female gamete. The female gamete, that is the ovum. Okay. Let us write the first difference between the two, that is the male gamete. The male gamete or the sperm is what? Produced in the testes. Produced in testes. And this is produced where? In the ovaries. Produced in ovaries. Okay, produced in ovaries. Let us see the second question. The second question is, the male gamete or the sperm will take a period of 64 days to mature. Okay, maturation takes place in how many days? 64 days. And for this one, the maturation will take place years. Maturation in years. Maturation in years. Okay, very, very, very important. The next, this will be motile. This will be motile, and this one will be non-motile. The male gamete or the sperm will be motile, and the female gamete, okay, I think you cannot see this, so I'll write it down again here. Okay, the third point I'll write down again. So only two I have to write at one go. So the third one I'll write here, this is motile and this is non-motile. That is, can move, and this one cannot move. The ovum cannot move. Then the fourth one I'll write that millions of sperms are produced per day in humans. Millions of sperms produced per day in humans, and here only one ovum is produced every 28 days. Only one ovum per 28 days. Okay, millions of sperms produced per day in the male. In the female, only one ovum per 28 days. Right? The last one, the last point, again I'll delete this, sorry, rub this. The fifth one, the body of the sperm, the body of sperm differentiated into the head, the neck, middle piece and tail, the middle piece and tail. Okay, the body of the sperm. Over here we see that the body, no differentiation of the body, no differentiation of body of ovum. Okay, no differentiation of the body of the ovum. So here the sperm body is differentiated into head, neck, middle piece and the tail. Here there is no differentiation of the body of the ovum. And the last point is reserve food material is res. Reserve food material. Okay, again this cannot be seen, so I'll write it again at the top. Reserve food material is res. And here I'll write sufficient reserve food material. Sufficient reserve food material. Okay. The reserve food material is res, and here there will be sufficient reserve food material. Okay. So with this we have completed this question. So what are the differences we saw here basically? Once again, can we just go through them? The male gamete will be motile, the female gamete will be non-motile. The male gamete, about millions of male gametes, that is sperms, are produced per day. Only one ovum is produced per 28 days. In the male gamete or the sperm, again we see that the maturation takes place 64 days. The female gamete or the ovum, the maturation takes place years. Okay, the male gamete or the sperm, the body is differentiated into the head, the neck, the middle piece and the tail. The female gamete or the ovum, the body is not differentiated and there is not abundant reserve food material in the male gamete. However, there is enough reserve food material in the female gamete, right? So we are done with this now, and now we'll move on with the next part. That is the next question we'll discuss. Describe the process of synthesis of fully functional mRNA in an eukaryotic cell, or how is this process of mRNA synthesis different from that in prokaryotes? Right. The first part of the question, describe the process of synthesis of fully functional mRNA in a eukaryotic cell. So we already know that the process of synthesis of mRNA is called as what? Is called as the transcription. We know the central dogma of molecular biology, which says that from the DNA the mRNA is synthesized. Okay, and this process is called as transcription. This process is called as transcription, where from the DNA the mRNA will be synthesized, and from the mRNA the proteins will be synthesized, and this is the central dogma of molecular biology. Central dogma of molecular biology. Okay, the central dogma of molecular biology. Now we see that from the DNA, the mRNA is being formed by the process of transcription. Describe the process of synthesis for fully functional mRNA in an eukaryotic cell. Now let us see the steps. So what happens, I think we have already discussed or seen in one of the question about the transcription unit. Okay, let us quickly describe. I'll just write in steps which you can probably write in sentence frame later on. So let us take this, that this is A only, that this is the template strand. Okay, this is the template DNA strand, template DNA strand, and this is the coding DNA strand. Okay, coding DNA strand. Okay. Now in the template DNA strand, we can write the direction from 3' to 5', 3' to 5', and this can be 5' to 3'. So what will happen, there will be a promoter region over here, the promoter region over here, and at this promoter region, okay, at this promoter region is what? Here the transcription will start. The transcription will start, that is the formation of mRNA will start. So to start there has to be an enzyme, so enzyme called as RNA polymerase. Why RNA polymerase? Because as I told you in the central dogma of molecular biology, from the DNA, the messenger RNA, mRNA has to be formed. So to form the mRNA, an enzyme called RNA polymerase has to be there. This enzyme RNA polymerase will bind to this DNA, that is the template DNA strand. There are two strands of DNA, the one strand which is being duplicated, this strand from which the the complementary mRNA will get formed. This strand will be called as a template DNA strand. The other strand is the coding DNA strand. The formation of mRNA on the template DNA strand always takes place in the 3' to 5' direction. Got it? Now what happens, in the template DNA strand there are three regions. One is the promoter region, the region at which the transcription, that is formation of RNA, starts. It starts at this region. Why? Because the RNA polymerase enzyme will bind to the DNA template strand at this point, and hence the transcription will start. Then we have something called as the structural gene. And this structural gene on the template strand will code for the proteins. And then finally we have the termination site, okay, the termination site, okay, on the template DNA strand, where this formation of mRNA, that is the transcription process, will end, and at this site the RNA polymerase enzyme will detach from the template DNA strand. Got it? So this is how the mRNA is being synthesized. Now this mRNA formed is not the functional mRNA, okay, is not the functional mRNA. It has to undergo various processes, right? Fully functional, because what this is mentioning, describe the process of synthesis fully functional mRNA. The RNA formed here is not fully functional mRNA. So what happens now? What happens now? This mRNA, okay, this mRNA will undergo what? It will undergo capping, okay, and tailing, Capping will take place at the 5' end and tailing at the 3' end. Okay, this mRNA will undergo capping, okay, and tailing, and then again it will undergo splicing of the introns, okay, then splicing of introns, okay. What are introns? Introns are that part of the DNA that do not code for any protein, okay, that do not code for any protein. And then ligation of the exon. Exons are those part which are undergoing, you know, these are those part of the DNA which code for protein, and then ligation of exons, okay, then ligation of exons, then ligation of exon, and hence, okay, and this will as a result will produce the mRNA, fully functional mRNA after this. After both these processes, that is, the mRNA undergoes capping at the 5' end, it undergoes tailing at the 3' end, you know, then the splicing of the introns and ligation of the exons, okay, and then finally the fully functional mRNA will be formed. So this is how, you know, describe the process of synthesis of fully functional mRNA in a eukaryotic cell. The second question B is the same thing, but there is a difference. It is asking that how is this process of mRNA synthesis different from that in prokaryotes. Okay. So the mRNA synthesis is different in prokaryotes in some ways. First of all, in the prokaryote there is a polycistronic DNA, a polycistronic DNA. Secondly, what happens here? There is no formation of, you know, there is no formation of the this thing, the exons over here. Thirdly, we see here that, you know, see the capping, the tailing, the capping, the tailing, the, you know, then the splicing of the intron and the ligation of the exon, okay, ligation of all these are what? All these are taking place after translation. All these are taking place after the mRNA has been formed. So these are the post transcriptional modification, PTM we can say that. Here in the there will be no, okay, here there will be no PTM. How that is, there will be no post translational modification here. Got it? Also here we see that the translation process is not separate, it is the same process. The processes of transcription and translation are not different. Okay? So how is this process of mRNA synthesis different from that in prokaryotes? So what do we see here? Once again I'll read this. How is this process of mRNA synthesis different from that in the prokaryotes? So here there is no post translational, post transcriptional modification. Okay, here there is no post transcriptional modification of tailing, capping, okay, tailing, capping, ligation and splicing. These things were present where? These things were present in the prokaryote, however this is absent over here. Also we see it is polycistronic. it is polycistronic DNA which is involved here. Okay, the polycistronic DNA which is involved here. Right? Polycistronic DNA where? The polycistronic DNA is involved in the prokaryotes. Okay? I'll write this as the polycistronic DNA is involved in prokaryotes. Right? The polycistronic DNA is involved in the prokaryotes. Also, there is no concept of intron and exon in prokaryotes. Okay? Also, there is what is this written? How is this process of mRNA synthesis different from that in prokaryotes? Also, no concept of intron and exon in prokaryote. Okay, can this be seen here? Also, there is no involvement of intron and exon in the prokaryote. Okay, I think it cannot be seen, so probably I'll rub this. No concept of intron and exon in prokaryote. So, two things: there is no concept of intron and exon in prokaryote, and also there is no post transcriptional modification over here seen in these cases. Okay, got it? So, with this we finish the first part of the question related to transcription. Next question: write the factors responsible for the depletion of ozone in the stratosphere. We all know CFC, the chlorofluorocarbon, which is given mainly from the refrigerator. Okay, refrigerator is responsible for the depletion of the ozone in the stratosphere. Right? Do you know the different layers of the atmosphere of the the stratosphere, the mesosphere, the ionosphere, and all that? Right. So, how does this affect the human life? If the ozone layer is not there, then what will happen? The ultraviolet rays will directly affect us, causing dermatitis, causing skin cancer, causing the opacification of the cornea, and so many things related to skin, and it can cause mutation leading to cancers. So, that is why the ozone layer is very, very important. I'll write this down. The the depletion of the ozone, the depletion of ozone is due to enhancement of what? Of CFC, that is chlorofluorocarbon. Okay, the depletion of ozone is due to the enhancement of chlorofluorocarbon, that is CFC. Now, what is happening? Because of this chlorofluorocarbon, the ozone layer is getting depleted. The ozone layer is getting depleted because of CFC. Ozone layer is getting depleted. and if the ozone layer is getting depleted, what will happen? The ultraviolet rays from the top will directly affect us. Okay? Because of CFC, the ozone layers is getting depleted, and because of this, what will happen? Due to this depletion, the ultraviolet rays, the UV rays will directly affect us. Okay? The UV rays will directly affect us, and when the UV rays will directly affect us, what will we see? Okay? When the UV rays will directly affect us, what will we see? We will see that the UV rays will, that is what it is asking, isn't it? How does this affect human life? This UV rays can lead to dermatitis, okay, the skin problems, opacification of the cornea, and other health related hazards, okay, and other health related hazards. So this is the responsible, why this ozone, why the CFC, is responsible for depletion of the ozone and what effects it can have on us. Let us now discuss the other question. Explain the biomagnification of DDT in aquatic food chain. How does it affect the population? Biomagnification is a concept, okay? Biomagnification is a concept where a material, a toxic material, it increases, its proportion increases as we go higher up the trophic level or the food chain level. So biomagnification, here the concentration okay, here the concentration of toxic material, okay, here the concentration of the toxic material increases as we go higher up the food chain, or the trophic level, okay. So here the concentration of the toxic material increases as we go higher up the food chain or trophic level, because these toxic materials, okay, because these toxic materials can neither be excreted, okay, or metabolized. Right? So biomagnification is a process where the concentration of toxic material increases as we go higher up the food chain or trophic level, because these toxic materials can neither be excreted or metabolized. And how does it affect the population? Yes, for example, let us take the example of a fish. See what happens, see in water the concentration of say any toxic material, say DDT, will be less. So when the fish or the any aquatic animal eats this fish, okay, say the the zooplankton, which is an animal in the water body, will eat this DDT. So it will consume, say it is in 0.2% per million, right, 0.2% per million. Now what will happen, this zooplankton will take up this what, this 0.2, it will take up this DDT, which is a toxic material. This zooplankton cannot excrete this, neither it can it can digest this or metabolize this. Then what will happen, okay, so it will remain in the zooplankton body. Now a fish, a big fish comes and eat this zooplankton, it will eat many zooplankton, so the bodies of all these zooplankton are having 0.2% of DDT. All this will get inside one fish, the body of one fish. A bigger fish eats the smaller fish, many smaller fishes, all this DDT gets again into the bigger fish. So as we are going up the higher trophic level, the concentration of of DDT or this toxic substance is increasing, and this is biomagnification. A human being cuts this big fish which is having many DDT and eats. So ultimately that DDT which was there in water in less concentration has reached a higher trophic level, the humans, in a very high concentration, and this is what is biomagnification. Okay, you note down this, and after this we will continue with the last part of the session. Okay friends, so I hope you must have noted down, and now we will continue to the next question, which probably is the last part of the section. Okay, define the following terms: acrosome, amniotic sac, birth, blastocyst, cervix, clitoris, and the coitus. So let us do one of them, one in detail, one by one in detail, not very much in detail, only 1 or 2 lines will quote about this. So the first one is what is the acrosome. The acrosome is the that part of the sperm which contains an enzyme, okay, which will enable the sperm to enter into the egg cell. The front part of the sperm which contains an enzyme this enables the sperm cell to enter the egg cell, right. This enables sperm cell to enter egg cell, right. So this enables the sperm cell to enter the egg cell. This is the acrosome. The next thing is the amniotic sac. Now the amniotic sac is a sac all around the embryo, which is protecting the embryo, acting like a cushion, so that it is protected from from jerks and so many things. So we can write this as the second point, amniotic sac. Amniotic sac. This is a fluid filled sac, okay. This is a fluid filled sac around the embryo, okay, protecting the embryo from jerks, protecting embryo from jerks and acting like cushion, and acting like cushion. So the amniotic sac is a fluid-filled sac around the embryo, protecting the embryo from jerks and acting like the cushion. Okay? Now we'll see the birth. The birth is actually a process in which, in which the new one, or the fetus, or the embryo, whatever you can say, from the uterus it gets into the new world. Okay, so I'll write this as birth process in which the fetus moves from uterus to the new world. Okay, from uterus to new world. Right? Then we'll see the blastocyst. The blastocyst is actually, a stage of the zygote. At the 9th day, at the 9th day, they say 5th to 9th day is the average day, but is the range, but the average we take as 9 day. Okay, but not average, because average will be lesser. Generally we take it as 9th day. At this stage of the embryo, at this stage of the zygote, okay, that is the 9th day, what happens, this gets implanted into the endometrium. Implantation of the zygote takes place at the blastocyst stage. So blastocyst, the blastocyst, okay, is this is a stage of zygote. This is a stage of zygote on 9th day, when it gets implanted, in the endometrium layer of the uterus. Okay, in the endometrium layer of uterus. Okay, so this is the stage of zygote on the 9th day, when it gets implanted in the endometrium layer of the uterus. Got it? Now cervix. Cervix is the neck of the what? The uterus. So I can write this here as D. Cervix is nothing but lower part or neck of uterus. Okay, when we draw the female reproductive system, this is, if we represent this as the ovary, okay, and we draw it like this, okay, this can be seen, right? So this is the fallopian tube, this is the ovary, this is the uterus. Okay, this is the uterus. This part, that is the neck part of the uterus, will be called as the cervix. After this will be the vagina, the vulva, and so on. So this is the lower, the neck part of the uterus will be called as the cervix. Then the next question, the clitoris. This is the sensitive part of the female reproductive system, that is the vagina. So this was E and not D, sorry. This is E. And finally we move on to X, F the clitoris. This is clitoris is a small sensitive part in front of the vagina. Okay, small sensitive part in front of vagina. Okay, this is a small sensitive part in front of the vagina. And the coitus, we are done with this. Coitus is a synonym for sexual intercourse. So G, I'm writing it here. Okay, coitus is the synonym for sexual intercourse. Right? So friends, we are done with this question. The acrosome is a small, small part in the front part of the sperm, which contains the enzyme for, for entering into the egg cell. Amniotic sac is the sac-like structure around the zygote or the embryo, which provides protection and has the amniotic fluid, amniocentesis and all. Okay, when we take out that amniotic fluid, that is by the process of amniocentesis, we can check for the, the sex of the fetus and also any genetic abnormalities. Right? Birth, it is giving, birth. What do you say? Birth. It is giving. Not rise, it is giving the birth to a new one. Okay, blastocyst, that stage of the embryo, the zygote, that stage of the zygote at the 9th day, when it gets implanted, at the endometrium layer of the uterine wall. Cervix is the neck or the lower portion of the uterus. Clitoris is the small sensitive part in front of the vagina, and coitus is nothing but the sexual intercourse. So friends, I hope this was a great, session. See, although we discussed 26 questions and so many questions we discussed in this sample paper, but I have covered the concepts apart from the questions, the concepts which are surrounding them. So all these 26 questions must be clear, thorough, logically, and I hope you learnt a lot from these sessions. Go back, study them, revise them, and probably we'll meet again with the next sample paper and some more sessions. A very good day, have a healthy day, a sound sleep, and keep up to your studies. The time we meet again, a very goodbye from Dr. Ashmir. Thank you.